All right, so day two on DNA notes. So today we want to look a little bit more at the structure of DNA and we want to go through the process of how DNA is able to replicate itself. So yesterday we talked about some scientists. We talked about Rosalind Franklin and Watson and Crick and Beadle and Tatum and Avery and Griffith and so on. So those scientists played an important role of discovering what the structure looked like, discovering how genetic information gets passed from one organism to another, and kind of leading up to this whole process of protein synthesis, which is going to rely upon DNA. So let's dive a little bit more into DNA itself. So we talked about Watson and Crick once again, and we said that they discovered the structure of the DNA molecule, but not without the help of Rosalind Franklin, who did the X-ray crystallography, did the view from above it, was able to see that it was a double helix. Like I said, she passed from cancer, most likely from the X-rays back then, and was never awarded the Nobel Prize like Watson and Crick were. So they say it's this double helix, which means that it's just this twisted ladder shape. And it's going to play an important role because it's twisted ladder and it's wrapped around these giant proteins called histones. We're able to fit a lot of information in a very small spot, so it kind of coils around. It's kind of like taking a little thread from your shirt and just kind of pulling it out, and eventually you get this super long thread. It's kind of how our genetic information is. We're able to kind of combine it and kind of put it into a very small spot, fit a lot of information, excuse me, into those particular chromosomes that this DNA is making up. So we know that if we take that double helix and we break it down into the small structure that we have, it's going to be made into a unit called a nucleotide. Know what a nucleotide is. We just got done with enzymes. We said, hey, a nucleotide is a sugar, a phosphate, and a nitrogen base, such as ATP. Nucleotide is also what makes up DNA, so it's made up of three components. Still, it's made up of a sugar. Here, this is a 5-carbon sugar. We talked about it a little bit yesterday with 5' end and 3' end. My sugar in DNA is deoxyribose. So even if you're like, oh, I don't know what deoxyribose is, it ends in OSA. You guys should realize that that ending gives you a sugar. We're going to have a phosphate group, and we're going to have one of 4 nitrogen bases in DNA: adenine, thymine, cytosine, and guanine. And we talked about these a little bit yesterday with Levine and Chargaff, and we said that with these particular bases, they form purines, which are your larger base pairs, double ring structures such as adenine and guanine, or single ring structures, thymine and cytosine, which are called pyrimidines. So when you take those nitrogen bases and we make those rungs of the ladder, we're always going to join one purine, double ring structure, with one pyrimidine. So your rungs of your ladder are always going to be 3 rings across. I can't put 2 purines across. That rung was going to be too wide with 4. I can't put 2 pyrimidines together. It's going to be too narrow, so I'm only going to have 2 rings across. So we have 3 of those rings, 2 of them from a purine, 1 of them from a pyrimidine. So the 4 bases that we had, those nitrogen bases, are going to allow us to have 4 different nucleotides. So sugar phosphate adenine, sugar phosphate thymine, guanine, cytosine, so on. When we get to RNA and transcription and translation, you're going to see that that thymine is going to get replaced by uracil. I know we kind of mentioned that along the way when we did organic a little bit, but this is where we're going to see it just a little bit more and the roles that that's going to play in that particular structure. So we know that these nucleotides are going to bond together, and they're going to form this double helix structure. So my sugars and phosphates are going to bond along that backbone. My nitrogen bases are bonding by those hydrogen bonds. So the 2 strands, now I've got this ladder, now they're going to start twisting. And there's a whole bunch of proteins or enzymes that are going to help with this particular process that we need to kind of unwind it. We need to open up those hydrogen bonds, and we need to replicate it. And that's what we're going to look at within this section of our notes. So once again, we're going to be kind of going back and forth, and you guys are going to see it in words. We'll see it in diagrams, and then we'll do a little bit of diagrams with what is known as Meselson and Stahl. We talked about them yesterday as being semi-conservative replication, so we'll apply it here a little bit more. Yesterday we went through the backbone of the ladder. We said that it is alternating sugars and phosphates all the way down. So sugar, deoxyribose, sugar, phosphate, and that goes along both sides of my DNA ladder. My rungs of the ladder, kind of where you put your feet if you're walking up a ladder, is the one that's going to be made up of the nitrogen bases. So these rungs of the ladder are kind of that variable portion that contains the genetic code. A, T, T, A, C, G, whatever letters that you have because of those nitrogen base pairs and the way that they're arranged make you who you guys are. Makes the code for your proteins, makes the code for all of your traits, makes the code for all your enzymes. and we're going to be looking at them in groups of 3, whether we're dealing with DNA, whether we're dealing with messenger RNA, whether we're dealing with transfer RNA. They're going to be in groups of 3. In DNA, this code, this grouping of 3, ATT, CCG, GAT, whatever it may be, this group of 3 in DNA is called a triplet. It's going to change a little bit when we get to messenger RNA. You guys might remember the word codon. When we get to transfer RNA, it's going to be called an anticodon, and my base pairs are going to change just slightly. So yesterday we talked about purines and pyrimidines. I said you guys will see it more in your notes, so here they are. Purine double ring structure, adenine and guanine. Your pyrimidine is that single ring structure. And the next page is going to have a little diagram. We'll look at it again and see how we can identify which is which there. We also know that adenine is bonded to thymine, so it's one purine, one pyrimidine, and that is with 2 hydrogen bonds. And then I've got cytosine and guanine, so my one purine, my one pyrimidine, and they have 3 hydrogen bonds. That way we know which base pairs are complementary together. So when this enzyme DNA polymerase comes in and reads one side of my ladder with base pairs there, he knows what base pairs get matched together based on these particular bonding patterns that we see. Yesterday, and I said, kind of let this sink in, we'll come back to it. We said that your DNA strand, this molecule, runs in opposite directions. We say DNA is anti-parallel. And we talked about this 3' end and 5' end. And this 3 and 5 is actually related to what number of carbon. Because remember, DNA has deoxyribose. It's a 5-carbon sugar. So we're looking at our 3rd carbon and we're looking at our 5th carbon, and that's it. Which carbon is facing up, our 3rd or our 5th? That's going to tell me what end we're dealing with. Down the bottom, is it my 3rd or my 5th? They're going to be opposite. So if this is 3' and 5', I know that this has to be 5 and 3. They will run in opposite directions. And it's going to be important when we go ahead and we replicate DNA, because DNA gets read, the old code, in one direction, and then the new strand is going to get made in a certain direction as well. So we say that the strand has directions between these 3 and 5 primes. So when nucleotides are joined together in the DNA strand, the phosphate group attached to the 5' end of the carbon of a sugar of one nucleotide links up with the 3' carbon of the sugar from the adjacent nucleotide. They are opposite. So the 5' end on one strand is going to match up with the 3' end on the opposite side strand, and the same for the opposite end. So here's my one side, here's my 3'. That means my 3rd carbon is facing towards the left. Up here, my 5th carbon is facing to the top. Opposite on the other strand, they are anti-parallel. They are running in opposite directions. So once again, the 2 strands are anti-parallel. They're extending in opposite directions, 5', 3'. So there's a lot going on in this particular diagram. You'll see it again in the coloring one here. So at the end, we'll kind of use it as a little bit of a review. So here's the backbone of my ladder. Here's my sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down. Other side, exactly the same thing. Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on. So basically, I've taken my double helix and I've stretched it out and made it into a straight ladder so that we can look at it a little bit more closely. These lines over here, this is just telling you what end they are, but we're going to look to see how we're able to determine it. Before we get there, let's look at the rungs of our ladder. So the rungs of our ladder are here in the middle. So if I didn't have these A's and T's and C's and G's in here, I might say, hey, go ahead and identify, and you may see it on one of the worksheets that you have. Go ahead and identify what you're looking at. How do I know which base pair is which? Well, we know here is a pyrimidine. It is a single ring structure. Here's a purine. It's a double ring structure. So great. I know that this is either adenine or guanine. I know that this is either thymine or cytosine. So the next thing you want to look at is the number of hydrogen bonds. And this is what Chargaff did. He said, every time I have adenine, I'm going to have equal amounts of thymine there. So here I've got 2 hydrogen bonds. So I know that these 2 hydrogen bonds join together adenine and thymine. If this is my purine, he has to be adenine. This is my pyrimidine, he has to be thymine. Going down, here's a purine, here's a pyrimidine. Guanine, cytosine. How do I know? I have 3 hydrogen bonds joining them together. So you should be able to identify things if I don't have those base pairs there, if you have the structures, if you have the number of hydrogen bonds. The next thing you're going to want to notice is that your nitrogen bases are always attached to your sugars. Always attached to your sugars. So my rungs of my ladder are attached to the side because nitrogen base pairs and sugars are attached. So far we've got what makes up the sides of my ladder. We've got what makes up the rungs of my ladder and how to identify all that. Now we want to look at that 3' and 5' end. So let's take this sugar right here. I'm going to kind of blow it up a little bit for you guys. We're going to take this sugar right here. It does not matter which one you pick. You might be like, hey, I want to take this one instead. Notice they're all exactly the same. They're running in the same direction. You will always start with your oxygen, and you're going to count clockwise your carbons. Where does that 3rd carbon end up? Up or down? Where does that 5th carbon end up? Up or down? And that's going to tell you the direction of that side of your DNA strand. So if I'm counting, and this makes it a little difficult because it's kind of understood that each little ending here is where I have a carbon. So when we looked at organic, remember I said that when you have that ring structure, carbons are kind of understood to be at those corners there. So there's my oxygen. I'm counting clockwise. 1, 2, 3, 3rd prime, or 3rd carbon is facing down, so my 3' end is facing down. 4 and 5, which is going to be right here at this little bend. So my 5th carbon is facing up. That means that this strand goes 5' to 3'. 3' at the bottom. Without even looking at the other side, I know it's opposite. If this is a 3' end, I know that this has to be 5'. If this is 5', I know that this has to be 3'. Let's check it out though. So if we're looking at that, once again, it does not matter which one you choose. They're all exactly the same. Because it's running in the opposite direction, you're going to notice that those sugars have kind of flipped over upside down. Because now my oxygens are facing down. So if I count my carbons clockwise again, 1, 2, 3, 3rd carbon's facing up. 4, 5, 5th carbon's facing down. Okay, so once again, my 3rd carbon is up. So this is my 3' end of my strand. This is the 5' end of my strand. And like I said, it's going to play an important role here in a moment as we go through and we replicate it. So if we think about DNA itself, so here's my DNA ladder that we're going to kind of unravel. If we think about how it all comes together, this DNA, like I said, has a lot of information. We got to kind of super coil it up, and there's going to be some enzymes that are going to help with that. But one protein that's going to allow this to happen is something called histones, which are like these giant balls that the chromosome or the DNA is going to wrap around in order to make that particular chromosome. So you can start to see that DNA double helix. It's wrapping around all these histones, and these histones are making up kind of like a big yarn piece of thread, and then that's going to be kind of super coiled together in order to make your chromosome. So a lot of information within these particular chromosomes. It's a lot of these DNA base pairs, A's and T's and C's and G's that are going on there. So Watson and Crick model, we said, of DNA suggests the shape of the molecules, double helix, that we know, 2 strands, double twisted ladder that we have there. But you guys should also realize that the 2 strands are complementary to each other. If there's an A on one side, I have a thymine on the other side. If I have guanine on one side, I have cytosine on the other side. They are anti-parallel. They're held together by those really weak hydrogen bonds. So on special occasions, this molecule is proven to have the capacity to copy itself. Well, it's going to need to copy itself. You guys have grown from an infant to a child to a teenager and eventually an adult. You need more cells. Well, within those cells is DNA. Well, in order to get DNA in those cells, we need to go through cell division. In order to go through cell division, I need to make copies of my DNA so that those cells have a copy of that DNA. So Meselson and Stahl, the 2 scientists that we talked about yesterday, proved that DNA replicates itself in a term known as semi-conservatively. That means that each strand that we produce, here's my old DNA chromosome, as it goes through and replicates, it's going to split, and each strand is going to be used as a template, and each strand is going to make 1/2 old, 1/2 new DNA. So I'm using my old strand as kind of like that model that I can build upon. Because remember, I have complementary base pairs here. Whatever's on here, I know what matches up with it. Whatever's on here, I know what matches up with it. So by the time I'm done, I have 2 strands that are completely identical. Well, now I have twice the amount of DNA within that cell. So what has to happen? Well, that cell needs to split. So now I have this DNA in one cell, I have this DNA in another cell. They are completely identical, unless, of course, there was a mutation that occurred somewhere along the line. So this is the idea of DNA replication. So when we talk about these different processes, you guys are going to see replication, you guys are going to see transcription, you guys are going to see translation. They each occur kind of in a different location. So remember, DNA, even though we think of this teeny tiny little molecule, it is a huge molecule. It cannot get out of the nucleus. So anything involving DNA, especially DNA replication, needs to take place in the nucleus of your eukaryotic organisms. If you're bacteria and you've got DNA, well, then you're going to take place in this little nucleoid area. You don't really have a nucleus, but you do have this nucleoid area. Even bacteria need to replicate their DNA before they go through binary fission and make two identical copies. So the process of copying or DNA replication takes place in all living things as well as viruses. So don't get confused. Viruses are not considered living. They do have DNA, and that's one of the characteristics of living things, but they can't replicate or produce more on their own. They need to take over our cells. So think about coronavirus. One person spreads it to another, it gets into your body, and then it takes over your cells and the machinery of your cells in order to make more of itself. That little virus has been sitting on a tabletop. It's not able to make more of itself on its own using the particles that it has. So we say it takes place in all living things as well as viruses. It takes place before cell division. It takes place during that interphase. So if you remember mitosis or meiosis, first year bio, cell cycle, we have interphase, G1, S, G2. We'll see a little bit of it as we work our way into embryology. G1, cells growing. S, synthesis. I'm replicating my DNA. G2, the cell finishes up, replicating all its organelles. Then it goes through mitosis. Prophase, metaphase, anaphase, telophase. At the end of telophase, I have two identical cells. So we said this process of cell replication, or this process of DNA replication, is going to take place during the S phase, the synthesis phase of interphase. In organisms that undergo sexual reproduction, meiosis. Because remember, mitosis makes identical cells. Mitosis is going through and making body cells. We're making identical copies, full set of chromosomes, half from mom, half from dad. Every cell that gets produced during mitosis is making body cells, full set of chromosomes, 46. When we go through meiosis and we're going through this process of sexual reproduction, we're producing sex cells, either egg or sperm, depending upon male or female. We're making cells that only have half the amount of chromosomes in them. Because egg and sperm for humans, we only want 23 chromosomes in them. Because when egg, 23 chromosomes, comes together with sperm, 23 chromosomes, we now create this little zygote, which will eventually form the embryo, which has a full set of chromosomes. We're still going to go through and replicate during that phase, but now we produce more products. So instead of producing 2 cells, we're actually going to produce 4 cells at the end because we're going to go through this division twice when we produce sex cells. We're not going to see that part here. We'll see it next when we get to embryology, but I'm just kind of putting in your head, it happens whether we're going producing body cells or sex cells here. We're always going through this process of DNA replication in order to make new cells. So DNA replication is proven to occur in 2-directional manner. So remember, we said that they are anti-parallel. And when I talk about this idea of semi-conservative replication, I kind of gave you guys like, hey, here's a quick little picture before we get into the really big diagrams of it. Here's my original DNA. I stretched it out. We talked about the enzymes that are going to do all this, your helicases and so on. And I have my sugar phosphate backbone. I have my nitrogen base pairs that make up the rung. I'm going to break my weak hydrogen bonds. This is my original strand. So it's kind of going to be like a little zipper that's going to go right up the middle and break it. When it does that, these 2 strands are going to split apart. And new base pairs are going to come in. So A goes with T, C goes with G, C goes with G, T goes with A. On the other side, I have the same matching base pairs. So by the time I'm done, these 2 strands are completely identical. Okay, each strand is made up of 1/2 old, which was your template, and 1/2 new DNA. The new DNA knew how to be created because it's reading the old DNA on both sides. However, they're going to be done in opposite directions. So one's going to get made in this direction, and one's going to get made in this direction. And that's where that 3 and 5' come into play. So this replication, once again, is going to occur during interphase. We said that there's 3 parts, G1, S, G2. S just means synthesis, to create. I'm creating new DNA. Semi-conservative replication, Meselson install, means that each strand contains 1/2 old and 1/2 new DNA. Once I have this, I now have twice the amount of DNA in that cell. That cell's going to need to go through cell division. It needs to divide. I can't have that much DNA in a single cell. So when that cell divides, I now create two identical, if we're talking about mitosis here, two identical cells, body cells. So each old strand, which are my blue here, are going to synthesize or create a new strand. So the new double strand is comprised of one old, which is my blue, and one new strand, and that was the mesylsin stall replication. So as we move from this diagram, because I don't have my 3' and 5' ends on it, to our next diagram, things that you need to remember. So DNA is kind of like a lot of memorization that we need to then apply to it. So base pairs, A and T go together, which ones are purines, which ones are pyrimidines, hydrogen bonds. You need to memorize that stuff. You also need to know that DNA gets read from 3' end to 5' end. It gets read 3' end to 5' end. I'm going to scoot back up to my last diagram. DNA gets read from 3' to 5'. So if this was going through and I was going to replicate this side or synthesize a new strand on this side, I'm going to go through and read in this direction. G goes with C, T goes with A, C goes with G, A goes with T. It's going to get made going up. This side is going to get made going down. So I would go, oh, here's one side of my old, T goes with A, G goes with C, A goes with T, C goes with T. So I'm going to keep going through. This one gets made in this direction, or gets read in this direction. This one gets read in this direction. As I'm reading it, I'm putting new base pairs in. As I'm reading it, I'm making my new sides. I'm making this pink side here. So as I'm reading my old strand 3' to 5', I'm making it 5 to 3. They're going to run in opposite directions. So you need to know DNA is read from 3' to 5'. And I'm going to do a whole poster project on my tabletop with movable parts for you guys and tape it and allow you guys to ask questions on that, because we used to do a big poster project that really helps solidify this information for you. But I'll do it on tabletops, allow you guys to kind of ask questions along the way with that as well. Okay. So we did this already. We've done it a few times. This is just giving you guys a little bit more help. So 3' and 5', how do you find it? It's the same diagram as above. I moved it over. There's my oxygen. I put your carbons in here. So if you have trouble seeing where those carbons should be located, remember I said at every corner here is where a carbon is. So this is really what this molecule looks like. So there's my oxygen. I count my carbons. 1, 2, 3. Bottom of the strand is a 3' end. 4, 5. Top of the strand is my 5' end. On the opposite side, same thing. I took this molecule, I filled in your carbons for you. 1, 2, 3. Third carbon's facing up. You're going to be like, well, so is the second. Yeah, but we're working with 3' and 5'. I don't care about the 2s and the 4s and the 1s. I want to look at my third carbon and my fifth carbon, and that is it. Which is facing up, which is facing down. So my strands are opposite. And I said, once you have one side, you're going to know what the other side is. They're opposite one another. If I don't give you guys the chemical makeup of it, and I just give you, hey, sugar phosphate, sugar phosphate, sugar phosphate, you're not going to be able to identify it. I have to at least give you the chemical makeup of it or give you one end of it. If I had just put a 5 here, you should be able to fill the rest of it in. Oh, if this is a 5' end, this has to be 3. They run opposite. If this is 5, this has to be 3. Strands are anti-parallel and opposite of one another. All right, so there's a lot to go on here. So when we go through DNA replication, what we're looking at is, first off, it's in a double helix. It's tightly wound. We got to kind of spread it out. And this process that you guys are going to see here happens multiple spots along the chromosome. It's not like we're going to find the end of our chromosome, kind of like the thread on a little shirt, and say, okay, let's start replicating from here and work our way all the way down the chromosome. It would take forever. So you're going to see, hey, it's happening here, 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 along that DNA strand, and eventually they just all meet up. So the first thing that we need to do is we need to uncoil this double helix, and we need to put it in that ladder structure that we keep seeing. So there are enzymes that are going to help to do this, and there are a ton of enzymes, and there are a ton more enzymes than what we're going to look at here, but these are kind of the main important ones that I think. Okay. Notice that a lot of your enzymes are going to end in ASE here. So our first one, the chromosomes are supercoiled. We need to uncoil them. And the first one that's going to do this into my ladder structure is topoisomerase. And it's an ASE. So topoisomerase is going to uncoil my double helix into my ladder. Once we have it unwound, we want to go through and we want to open it up. So the next enzyme that you have is this enzyme called helicase. And helicase is going to kind of break those hydrogen bonds and kind of open up that strand like a little zipper. So pretend there's a little zipper like on your coat and you're unzipping that coat and you're opening up those bonds. When we do this, notice that it's kind of creating this stretched out molecule here. This is known as a replication fork. I can't just say, oh, here's the replication fork, because it is constantly moving. As that helicase is opening bonds all the way up, that replication fork is moving up. So that replication fork is where the strand is opening. We have an initiation site. Initiation site is where replication begins. We have something called elongation. So my main enzyme, what I consider my main enzyme, once we have it unwound, once we have it opened up, we have my two original strands and they're in that kind of replication fork opened. We now need to have DNA polymerase come in and start adding new nucleotides, because you guys have all these free-floating nucleotides within your nucleus. Sugars and phosphates and nitrogen bases, they come from the foods that you guys eat. So you have them all floating around within your nucleus. DNA polymerase is going to start matching them together. He kind of deciphers the code on your original strand. So as he goes through, he's reading from 3' end to 5' end, and he's working his way. He's going, oh, there's an adenine. Let me call in a thymine nucleotide, sugar phosphate thymine. Let me bond them together. Hey, here's cytosine. Let me call in a guanine nucleotide. He is reading the one side and he's adding those new complementary base pairs in along the other side. So that DNA polymerase begins to read the DNA from 3' to 5'. He's working in an opposite direction. So as he's working up on one side, he's going to be working down on the other. And that's going to cause just a slight little problem there. And we'll see that on the next diagram. All right, so as they're going through and they're matching up those nucleotides, replication is going to continue. However, on the 5' side, we know we're going to see something called a lagging strand. That means where the 5' is at the top on that old strand and we're kind of trying to read it, it's going to be called a lagging strand. And the reason is because we're moving away from the replication fork. So what's going to happen is that DNA polymerase has to kind of reverse the direction it's making DNA and kind of begin at the 3' end. So what he needs to do is he needs to rely upon something called RNA primase. So you're going to go, wait a second, this is DNA. Why am I dealing with RNA primase here? RNA primase is just another enzyme. It's going to kind of come in and it's just going to add a little base pair there, and it's kind of like a little docking point. Because what has to happen is that my polymerase has to keep breaking off, going back up, reading, breaking off, going back up, only along the one side that you guys will see on my diagram here. So RNA primase is going to create kind of this little hook so that the DNA polymerase can attach to. Eventually that RNA primase piece that we add in there, it gets cut out. And then all my DNA parts come back together again. So even though I'm throwing a little bit of RNA in there, it gets cut out and my DNA molecule is kind of going to get glued together. So we say that it creates a little hook for the DNA molecule so that it can attach. Then the RNA strand is replaced by DNA. Cut out, put my right base pairs in. This process is discontinuous. So I have two strands that you guys are going to see. You heard about them in first year bio. It's called a lagging strand and a leading strand. And that lagging and leading strand relate to my old or my original strands of DNA that my polymerase is reading 3' to 5'. Because my lagging strand is going to be a discontinuous formation. It's going to keep going and it gets to the end. It goes, oh, I got to go all the way back again because my replication fork is constantly opening up. I'm going to create something called Okazaki fragments, only on my lagging strand, only the one being made away from the replication fork. The other strand is continuous. It's going to get read and it's reading and it's following my replication fork and it's happy. However, this one's going to take a little longer, my lagging strand. At the end, semi-conservative replication, I'm ending up with two strands that are completely identical. In order to glue those Okazaki fragments together, I'm going to rely upon one more enzyme, and it's called ligase. Ligase is kind of the glue. It's going to glue together those DNA portions, those fragments that I have on my lagging strand. So it's only used on my lagging strand because it's going to be discontinuous. So before we get to this diagram, one more enzyme called nicase. We said that this should be kind of foolproof. A goes with T, C goes with G. Yeah, we know that that's not always the case. Mutations can occur. So the DNA gets proofread for mistakes. And as errors are found, because remember, we have billions of these base pairs that are being added in there, the nicase is going to cut out the wrong base pair and put in the correct base pair. Is it still 100%? No, you still might have base pairs that just have slight errors within them. Maybe it means nothing. Maybe that portion of your DNA codes for nothing. Maybe it's nonsense DNA, which we have a lot of. So things actually are not always going to code for proteins and enzymes. You guys have a lot of junk DNA within your chromosomes as well. All right, so let's look at this real quick. This is semi-conservative replication. So you're like, oh, a lot going on. I know that. My blue strands, and we'll do it again on a couple of diagrams here. My blue strands are my original DNA. I don't see sugars or phosphates or nitrogen bases or any of that stuff. So I have to tell you, hey, here's a 5' end on my original DNA. By that, I know if that's 5', this end is 3'. If this is 5', I know the other side of it is 3' to 5'. The blue is my original DNA. So we had topoisomerase. It took that double helix twisted ladder and made it into a ladder. Helicase came in and broke my hydrogen bonds. And it's creating this replication fork. So right now, here's my replication fork. It's going to keep opening up. So it started way down here, and my blue strands, my original strands, started opening up. So I have all of my base pairs here. I have my complementary base pairs on this side of the blue. It's going to keep opening. So my replication fork is going to keep working its way up. This is happening multiple spots along the chromosome, and eventually they're all going to meet, so that entire chromosome is going to be replicated. All right, DNA is read. When we read DNA, it is read on the old strand that contains our code, your DNA, your code that makes everything about you. So DNA polymerase is going to come in and it's going to attach upon the old strand right here. It is reading 3' to 5'. So he's going to come in and go, oh, here's an adenine. Let me throw in a thymine. Oh, here's a cytosine. Let me throw in a guanine. Here is a guanine. Let me throw in a cytosine. Here is a thymine. Let me throw in an adenine. So he's going to be working up in this direction. It is reading 3' to 5'. My new strand, because remember those strands are anti-parallel, and had I give you the chemical makeup down here of my sugar, my 5th carbon would be facing down. It gets made 5 to 3. So notice my polymerase. My polymerase is opening or is working up towards the replication fork. It is continuous. Not so on the other side. On the other side, let's switch some colors here. So on the other side, what's happening? Well, my DNA still gets read 3' to 5', my old DNA. So let's pretend that my strand was kind of closed up to here and started right here. So here's my first. DNA polymerase comes in. He attaches to my old strand. He is reading 3' to 5'. He's reading in that direction. Polymerase comes in. He starts adding stuff in. He goes, okay, well, A goes with T. A goes with T. T goes with A. C goes with G. G goes with C. And he gets down to the bottom. He goes, well, now what do I do? He looks back, and the fork opened up more. So now he's got to go all the way back up. I'm going to switch colors just a little bit here. He's got to go all the way back up, and he's got to hook back up to the strand. In order for him to hook back up to the strand, dock onto that strand again, because he has to keep breaking off and hooking back on, that's where RNA primase comes in. He's kind of like that little dock on a lake, so you can hook your boat up to it. So that RNA primase is there to make that little docking point, so DNA polymerase can come in and attach again. So he attaches again, and he's going to keep doing it. That strand is still getting read 3' to 5'. And he's going to keep going. Oh, okay, T goes with A. G goes with C. C goes with G. A goes with T. And it keeps going. This strand is getting made in this direction. It is still getting made 5' to 3'. My strand is still getting read 3 to 5. So this strand is now discontinuous because my DNA polymerase keeps breaking off. He goes and does it again. He goes, oh, okay, I got to go back. My replication fork opened up again. Let me hook on to where that RNA primase made my little docking point. He hooks back up again, and he's going to keep going. By the time you're done, this strand and this strand is going to be completely identical. This strand takes a little longer to do. Because of that, this strand is known as your lagging strand. This strand is known as your leading strand. It's leading because it's leading the way. It is following the replication fork. It doesn't have to break off. DNA polymerase is happy. A goes with T, T goes with A, C goes with G, and as that strand opens up, he's following it. On this side, he's lagging behind. He gets down to the bottom. He looks back. The fork opened up more. He's got to go back, hook back on, do it again to where he caught up with it, go back, do it again, and it takes a little longer to do. But the end product is completely the same. Because on my lagging strand, we keep breaking off, I have my first fragment here, my second fragment I did in purple, my third fragment I did in blue. Those are called Okazaki fragments. They will get glued together by ligase. So ligase is that enzyme that's gluing everything together there. So that at the end, this strand and this strand is completely identical. Meselson and Saul, semi-conservative replication. So if we look at these diagrams, and this is the one that we did when we did our scientists, and we talked about Chargaff, and we talked about Levine, and how you guys can identify your sugars and phosphates. So if I gave you something like this, and I said identify a 3' end and a 5' end without these numbers here, you would never be able to do it. So I would never do that to you. If I gave you one end of it, you would then know how to do it. Oh, this is 3', they're opposite, that's 5'. If I follow him all the way down, and so this end has to be a 3' end because they're opposite, which is exactly what it is. We said that they run anti-parallel. So if we look at this replication here, so here's my original DNA. Let me see if I can kind of give you guys my original DNA here. So here's my original DNA. I'll put it in yellow for you. All the way down, as well as this strand. So here's original DNA. We're untwisting it. Topoisomerase is kind of untwisting it for me. Start this color a little bit. And helicase has gone through and broken it apart. So there is my original DNA. I am now going to go through and I'm going to start adding some new DNA in it. Okay, so down here, I told you which ends are my 3' and my 5'. This original DNA, my yellow strand, my 3' end is at the bottom. So this DNA is going to get read in this direction. So it's going to get read in 3' to 5'. That right there, I know, is my leading strand. He is following my replication fork. Where's my replication fork? Well, right now it's right here because that's where my original base pairs are still together. But I also know that helicase is coming in and kind of breaking those base pairs. Breaking. So my replication fork is going to keep moving. And once again, this is happening in multiple places along the way. These big circles that you guys see right here, this one here, that is my enzyme polymerase. And this one here. So that is DNA polymerase. That is the one that is reading my old strand and telling which ones to come into play here. So we just said polymerase started on this side, polymerase started down here. And he started reading. Hey, here's a cytosine, let me add guanine. Here's an adenine, let me add thymine. Here's a thymine, let me add adenine. Here's guanine, cytosine, and he's going to keep going. So he's adding adenine, cytosine, thymine, guanine, adenine. He's going to keep working his way up that particular strand. So he is reading it 3' to 5'. He is making it 5 to 3. So the top of this strand is now going to be a 3' end. On this side, if we're looking at it and we go through, down here is a 5' end, so that I know that if I go all the way to the top, I need to start up here. DNA is going to start reading it down this end here. So we're going 3' to 5'. However, we can't start here because that's not where I'm opening up. So I'm going to slide all the way down. And let's say my strand was only open to about right here at that particular point. Maybe it was open right here. So what happens? DNA is going to come in. It's showing you right here. So we'll start here and it's going to start adding them in. So it's reading 3' This is a 3' end over here because this is a 5' end. It's making it 5' to 3'. So polymerase goes, hey, here's C, let's put a guanine. Here's thymine, let's throw or adenine, let's throw a thymine. Here's guanine, let's throw a cytosine, and it's working in this direction. He gets down to the bottom. He turns around. He looks up. My replication fork opened up. He's got to come back in. He's got to attach back on. He's got to do it again. So this side here is known as my lagging strand. They're both going to be the same by the time that we're done, but I'm going to have to utilize a couple more enzymes here. So RNA primase will be on my lagging strand because it has those little docking points for polymerase to keep docking onto as it keeps breaking off. Because remember, this polymerase, he's just following the replication fork. He's just chugging along, adding those base pairs, and he's going to be done a little bit more quickly. He has a lot more work to do on this side, this polymerase. He gets to the bottom, he looks back, replication fork opened up, he goes, oh, goes back up, attaches onto the strand where the RNA primase made the little hook or docking point for him, does it again, makes another fragment. Those fragments are Okazaki fragments. All those fragments are going to get glued together by the enzyme ligase. By the time that we're done, these lagging strands and leading strands are going to make two new strands that are completely identical. Now that cell has twice the amount of DNA in it, and we're going to go through and have cell division. Mitosis, meiosis, nucleus, we're dividing it. This is all happening during interphase, during the synthesis part of the interphase portion of it. Underneath, you guys will also see another diagram. It is the same thing that we kind of just went over. It's the same exact thing that we just went over. It's just a different view. Okay, so I do like this one because it shows you all your enzymes together in one place. So we had this super coil up here. We said that, topoisomerase is going to uncoil that DNA. Your helicase is breaking. Notice the little arrow right here. It's working in this direction. So there's my replication fork, and helicase is breaking those original arrows or breaking those original base pairs. My polymerase is going to come in, number 3. You'll see it on both sides. And it's going to be working. So notice, you can't always say, oh, my leading strand is always going to be on this strand, and my lagging strand is always going to be on this strand. They're switched here. You can't do that. You have to look to see. DNA gets read, the old strand, 3' to 5'. So it is going in that particular direction, 3' to 5'. It's being read in this direction here. This DNA is being read in this direction here, 3' to 5'. DNA gets made, 5 to 3. It is getting made in this direction. It is getting made in this direction. They are anti-parallel, is what you guys are seeing here. So once again, topoisomerase is going to uncoil. Helicase is going to break those hydrogen bonds and create the replication fork. DNA polymerase, we're going to see on both sides. It basically is going to join or add in those new nucleotides that you have floating around within your nucleus. On my leading strand, it has an easy job. It's following my replication fork and it's working its way up. On my lagging strand, you see I have a bunch of fragments here already. So polymerase attached here first. It went through, made a strand. It broke off, hooked onto the RNA primer, little docking point, made a new little segment or fragment there. Broke off, hooked onto the RNA primer, made a new little fragment. Those are my Okazaki fragments. They will be glued together by ligase. So here, this is my leading strand, this is my lagging strand. By the time we're done, they are two identical strands. Your last enzyme is going to be nicase, and nicase is going to kind of go through and proofread. Not always 100%, but definitely will get rid of any errors that we have there in case an adenine pairs with a guanine, where we go, oh, that can't happen. It's 2 purines and different number of bonds. Anything's kind of able to happen within this particular structure. So it will go through and kind of proofread for you guys. Okay, so that's DNA replication in a nutshell, kind of a very large nutshell. Stuff that you guys have seen for first year bio, probably a lot more with the enzymes that we have. And like I said, this is just scraping the barrel layer of what we have in terms of enzymes. There are a ton more enzymes that are involved with these particular reactions. There's so many reactions that are occurring here. Probably things that we still haven't even discovered. So DNA, Nobel Prize, we're looking in 1960s. That was about 10 years before I was born. So to me, I think about it, I'm like, holy mackerel, think about how far we've actually come. I think we've cloned organisms, genetically modified organisms within a very short period of time. So within your lifetime, what we're going to be able to do with DNA and maybe hopefully go in, and we're starting to do this now. If you guys have heard the word CRISPR before, it's kind of this gene altering program that they're working on. Can they go in and kind of fix gene mutations there? Can we get rid of different genetic mutations along the way? And that's something that they're really working on. That's kind of the new thing. So if you're like, oh, I haven't really heard of GMOs, GMOs are old, genetically modified organisms, and we'll talk about them, and we'll talk about CRISPR a little bit. But this process is just moving at a very quick pace. Okay, so that's DNA replication. We're talk a little bit about restriction enzymes on the next time we see each other, and we'll also start breaking our way into protein synthesis. Why does DNA have to go through and rely upon RNA? Okay, so once again, as you guys go through this particular thing, you guys can repeat, you guys can go back, watch little parts of it, write down questions. Holy mackerel, I have no idea when you're talking about 3' 5' compounds. I don't understand leading and lagging strands. I don't understand what topoisomerase is doing. Write down questions so that we can go through it. I guarantee you, if you have a question, somebody probably has the same question. Please, please, please ask. Okay, so that is it. Thank you.